Hello?
I can hear you.
Okay, sorry.
I can't see you, but I can hear you.
Yeah, sorry for the delay. Yeah, I just wanted to announce that for the fair, we are having a photography contest and an art contest. As well, for the day of the fair, we're going to have a costume contest for the first time ever. So as far as the art contest and the photography contest, submissions are going to be via email. I think we should be sending out those flyers via email, but they're on our Instagram page right now. And then for the costume contest, the judging will take place the day of the fair. So you would just wear your costume to the fair and have it judged there. But yeah, just wanted to make those announcements in case you have any mushroom photographs or mushroom art that you'd like to show everyone. The winning photographs and art pieces will be displayed at the fair. So if you want a chance to have your art displayed at the fair, this is a really good opportunity.
It sounds great. Well, I'm excited for the mushroom costume contest. I think that's going to be pretty interesting.
Yeah,
so
I would think.
I'm glad we're offering that to our members, the opportunity to do that.
Yeah.
Yeah, I heard people wanted it, so I was like, okay.
Yeah, that's a really good idea. Good for you. And Vanessa, I'd like to ask John too. So John Thrope works with Vanessa pretty closely to make sure that we have the volunteers that we need for the fair. So John, do you want to come on and say anything about our volunteer status?
Yes, I have received everyone's email. So if I haven't gotten back to you, it's because we don't have the schedule ready yet. So keep an eye out. We're going to send out a mass email within a couple of weeks, and everyone will get to choose their preferred station and times.
Well, that's great. I'd like to thank everyone for volunteering. The fair cannot happen without the volunteers. There's actually a lot of human power and thought that goes into setting everything up and taking it back down. And thanks to John for coordinating with Vanessa on that. And thanks to all the people who have volunteered so far. And if you're interested in volunteering, John's contact information is on the LAMS website. And so it's not too late to get in touch with him. Do we have any other LAMS business that we need to discuss? Well, if not, I'll go ahead and turn it over to Aaron Thompson, who's going to introduce tonight's speaker. Aaron?
Awesome. Thank you, David. I'll just plug real quick the reading group. We'll be finishing up Finding the Mother Tree by Suzanne Simard in January. So if you're interested in reading and discussing that with us, there'll be some announcements on the 2nd and 4th Monday of January. So onwards, I'm really excited to introduce Dr. Kristen Wickert, hailing from Appalachia Country. Dr. Kristen Wickert studied forest biology at Penn State and then plant pathology at West Virginia University. Today, she is a professional naturalist who studies fungi, arthropods, and plants and how they interact and rely on each other for survival. She has published papers on all sorts of tree and bug interactions, as well as fungi, on native and invasive species as well. She uses this knowledge to help influence people's perspectives of nature and teach them how they are a part of a system and they can contribute to preserving the magical creatures that they share our home with us. As a professional educator, she teaches children, adults, and even politicians about how our actions affect a landscape. Dr. Kristen Wickert also does an incredible work in science communication with her brand, K-Doves, the hiking scientist. So follow her on Instagram I'll link it in chat and you can scroll through all of her foresty adventures. And on to that, I'll hand it off to Kristen.
Thank you. That always makes me so, I don't know why, embarrassed, but that's all. You said very nice things about me. All of it is true. I am in Southern Appalachia. I love it here. I used to live in California, and it's my favorite place to vacation, not live. So I'm actually going to be at Soma. I know it's in the north, but I will be at Soma in about less than a month. So I'll be back in the wonderful state of lots of driving and tasty food. I'm excited. But thank you so much for letting me come here tonight. I tried to make my talk slightly regional. It'll be very broad at first, and then it will also be catered to you guys down in Southern California. I also love Southern California. I've done a bunch of hiking down there near Tehachapi and Red Rock Canyon, and I have some little fun pictures from there. But I'm gonna share my screen, and it looks like it's working. So here we go. Ta-da! Okay, so I, like that awesome introduction that I'm still very flustered about said, I studied plant pathogens, which mostly I focused on fungi. So a lot of the pictures you're going to see in this presentation are mine from my experience of culturing fungi, and then also a lot of it will be directly from work that I did when I was younger and worked in a lab. So we're going to talk about fungi as plant pathogens today, and I'm going to advance it, and I hope it works. Is it working? Is it me as a glamorous young person? Okay, yes. So when we used to get dressed and go to work, I used to go into a lab and culture fungi that cause diseases on plants. And I have a bachelor's in forest biology, a master's in plant pathology, where I studied entomopathogenic fungi, which I would be happy to talk about another time for days because it's fascinating. And then I transitioned into a PhD in plant pathology as well, but within that, there was the facet of actual plant pathogenic fungi, since you'll see that everything is all connected. You can be an expert in one thing, but really you're like a pseudo expert in many things because they all work together, especially in this field. And because of that, I've had many jobs, and because of the economy and just being a millennial, I've had many jobs, and I have to learn everything. And just, you know, I can call myself a professional entomologist, mycologist, botanist, whatever the government wants to pay me to do, I'll figure it out. But I have a lot of different experiences with just general ecology. Some of my publications have included organisms in California, and a lot of them are the interactions of fungi with plants and also the interaction with insects. And sometimes those will all be together because you can't have an insect that transports a fungus that kills a tree without one of those pieces, right? So you have to kind of know the whole story if you want to know one organism in that story. So let's start with what is plant pathology. My mom doesn't even really know. She calls me a tree doctor, but I kill trees. But in general, the field of plant pathology is the study of organisms and abiotic stressors that cause disease to plants. And this is a perfect picture over here of a soybean leaf that has symptoms of a disease, right? There's the yellowing chloroticness, and there's the brown necrotic area, which means it's dead. And it's causing a disease that will eventually probably kill that whole plant. And there are many different types of plant pathologists. There are people who specifically study nematodes, which is that little beautiful roundworm down there. There's people who only know things about bacteria, and there's people like me who really, I know a little bit about some of those, but most of my experience is in mycology. And even within that, there's more specificness where you can be a plant pathologist that's a mycologist that only works on trees. I know nothing about soybeans, even though that's where all the money from the government goes in plant pathology, because we love our soy and corn. But I go out in the forest and I look for diseased trees and try to figure out what's hurting them. And like I said, a lot of being a plant pathologist is just making observations, trying to look for trees that are sick or other plants that are sick, and then killing those plants to figure out if it's really the organism that you think killing that plant. And this was first done professionally by a gentleman called Robert Koch, and he was a microbiologist. And so he has been accredited with these postulates that all plant pathologists use, called Koch's postulates, where you first make an association. And you will go out in the field and you'll observe a sick plant. And you can see there's this plant here that's supposed to be just a blade of grass. It's green, it's healthy, but there's these yellow and brown parts with some of these black dots. And you'll try to do the next step of Koch's postulates, which is isolation. So if I had this blade of grass here, I would take a sterile needle that I cleaned with bleach or alcohol or a flame, and I would scoop at those little tiny black dots because that's likely the organism that's causing that disease. And I'd put it on nutrient agar to try to get it isolated by itself. I would extract that causal agent. Then to see if that thing I extracted caused disease, I would have to take it from that beautiful little petri plate and put it back into the same plant that's healthy to see if I got the same results. And so that's what I did a lot of my research on, because in order to say this is a problem that needs to be taken care of by the government, the public needs to know to not transport it, you have to prove that that organism is killing that plant. And in order to finally end that step, you have to re-isolate the same organism out of a plant that is showing the same symptoms as the one you found outside. And here's a real life plant pathology example of that, where, I mean, diseases are being found all the time. This was a project that was maybe like 2017, so not too long ago, even though time flies now with coronavirus. But this is a plant on the East Coast where I live that's called Pyrularia pubera. Its common name is buffalo nut. It makes a really big nut, and it's a parasitic shrub. It's super cool. It's a parasite of trees. And it was seen in a forest not too far from where I lived before that had these big swellings that had never been seen before. So this shrub had these really gross necrotic areas, and it also had a bunch of little sapsuckers coming up. So these are all symptoms of a sickness. And then there were these oozing pustules. So we looked at those oozing pustules, sterilized a tool, scooped it, and then put it on other plants, right, to see if it would cause disease. So here is a close-up of those oozing pustules, which, spoiler alert, it's fungal tissue. So we isolated it onto plates where, you know, all we do is just hack it apart and put pieces of the wood on a nutrient agar plate, and the fungi will actually grow out. And you can see there's a bunch of different colors going on this plate. So this is the actual plate of this sick buffalo nut shrub. And there's some like whitish fungus coming out of it. There's some blackish fungus coming out of pieces of the wood, and then also some beautiful pink stuff. And so I had to isolate each one of those, all three of those different organisms, and then do the next step of putting them back into a healthy plant to see if they cause the same symptoms. And here are the results of that, where we took these fungi, put them into some stems, and you can see that some of them caused disease, whereas a control did not. So we were able to find a certain organism caused this disease in a forest in my neighborhood, and now is something that's monitored for by local government forest agencies. And I know you're probably all saying, where are these big charismatic mushrooms that we are all in a mushroom club about? Because that stick with those pustules isn't really too obvious. Well, most of those big, beautiful mushrooms that you find in your forest are in a different type of relationship with the plants they interact with. They'll be mycorrhizal or saprotrophic. They're not necessarily acting as plant pathogens. There are a few exceptions in the world of plant pathology where the basidiomycotin fungi, which I'm assuming you all know what basidiomycotin fungi means. Basidiomycotin fungi that act as plant pathogens are in the genus Armillaria, and also in several genera within the polypore group. So the honey mushrooms, which I know you guys have of different species than me, but those honey mushrooms, those are considered to be a secondary plant pathogen, and here they are acting as a rhizomorph stage killing a tree. And then if you ever see those conchs on trees, like a polypore in the genus Felinus or Ganoderma, you can assume that it is actually actively eating away the heartwood of a tree. So many of our plant pathogens are in a different phylum than Basidiomycota. They are in the phylum Ascomycota, and they cause vast amounts of disease to our agricultural systems and forest trees. And they actually get a huge amount of your tax money, because if they attack and kill food and we don't have food to eat, that's a problem, right? So here's just a little bit more information about what an ascomycotin fungus is, right? They are in a separate phylum from our big obvious mushrooms that we were just looking at in the email session of our meeting. And they have structures called cups, right, which are filled with ascus, and those have ascospores. So it's a lot of steps, but most of the plant pathogens have these sacs of spores like this, a lot like our morels and little cup fungi, like the scarlet cup or the pizzizias. And so we can grow these fungi on nutrient agar plates, and they'll still grow these reproductive structures of their spores, and we can identify them based off of that. There are a few more exceptions for Basidiomycotin pathogens. Our rust fungi are Basidiomycotin fungi, even though they look like Ascomycotin because they're just kind of fuzzy, they don't have the big mushrooms, they're not very obvious. But these Ascomycotin fungi are all around us on a daily basis, and a lot of them are acting as pathogens or deteriorating things. But you can, of course, say thank you to Ascomycotin fungi for your cheese, bread, and alcohol. Those are all responsible for the fermentation process, or the blue cheese. The blue part is actually fungi, and the rind of brie is fungi. You can thank Ascomycotin fungi for medicine, something that helped the United States and the Allied services win the war, where penicillin is made from penicillium, and it's a chemical that will kill bacteria that makes us sick. There's also the opposite end of that, where diseases on our body are caused by Ascomycotin fungi. So if you have itchy feet, you can say hello to your Ascomycotin friend that's always with you. And it's also the reason our produce goes bad. So if you ever have bananas and you see these little dots on there, those are infection points of a fungus called Calototrichum. And of course, there's the environmental molds that are in our house, just all over the place, and the leaves, whatever, that just look like the green, messy masses that grow all over the place. But we're going to focus on a really big group of the Ascomycotin fungi of the plant pathogens. But many of these plant pathogens are super, super cryptic, where you're not even going to be able to see them growing as a fuzzy mass on top of leaves. They're all internal, but you have to guess them based off of their symptoms, because they all kind of do certain jobs. So there might be a fungus that causes yellowing of soy, and then there might be a fungus that causes the, oozing pustule on a stem. But many of these are invisible to the naked eye, and we have to grow them on nutrient agar to identify them. So they'll grow out of a plate like this in a radial mycelial growth. And then we can use microscopes to look at their distinct spore features and the way that they grow. Here are some spores of a fusarium fungus that look like little canoes. And here, this is an entomopathogen that is it used to be called a different name that hinted at looking like grapes. And sometimes identification of plant pathogens requires patience. So this is all the same. I don't know why it's advancing. I'm so sorry. This is all the same fungus grown on slightly different nutrient agars, but it shows some of them can be really fuzzy on top and some of them can be really smooth, and they can look totally different. But that's, a result of heat. That's a result of food and sugar in the agar. It's kind of a wild gambit. So it really does take some skill to look at these fungi and identify them. But plant pathology has been around for a really long time, longer than us, but it's influenced us since our beginning ages of being civilized people. And here's a good example of a plant pathogen that is called wheat rust. It's just a rust fungus, and it destroys wheat crops. And so ancient Roman cultures actually made a god that was in charge of plant pathogens and if they were coming that year or not, right? And we all know that fungi really do well when it rains. Well, it would just be different years that there would be rain and then the wheat crop would go really bad. But they didn't understand that yet. So the only way that they thought that they could stop the rust fungus from killing their wheat was by killing virgins and animals. So it's been around and influenced a lot of our actions as people for a long time. Another story that's really famous in plant pathology is the story of St. Anthony's fire in the Middle Ages, where people were losing limbs and they were suffering from the effects of ergotism, where they would have convulsions, they would have hallucinations, and there were actually people walking around very commonly with these different structures to help them get around because their legs had rotted off. It would even happen to livestock. And that's because of a fungal infection of rye, which a lot of people in Europe were eating during that time. And it's a fungus called CLAVICEPS purpurea that replaces the grain with a fungal structure here called a sclerotium. And it produces these little stromata that make spores. But those little like fungal sclerotium also produce lysergic acid, which is a derivative of LSD. So all of these people were eating this when they would make bread, and it would transfer the chemical and cause them to have vasoconstriction. So blood wasn't getting to their brain, they were hallucinating. Blood wasn't getting to their limbs, they were losing their fingers because they had poor circulation. And this stuff still happens today. Fortunately for us, not in our country, but in a lot of poor countries, it still does, because this fungus hasn't gone away. It still occurs. You can see this is a picture, right, in color, really good graphics and quality, but we have these mechanical sifters now that can sift out these sclerotium from the rye grain, and we don't have the problem anymore of the ergot getting into our food. But in poorer countries where they don't have that ability of machinery, there's still cases of ergotism today. Another case that's pretty famous, it's not necessarily a fungus, but this is an organism that gets lumped into mycology often, is a water mold. So it's a mycototic organism, and it was responsible for the Irish potato famine. So it's still studied in the field of plant pathology today. It's different than a fungus in that its spores, which is similar to fungi, has animal-like a tail. So it actually will swim around and go and swim in water and cause new infestations. And Ireland was a perfect storm for this water mold infestation that killed all of their potatoes because of Ireland's habitat, right? Just like I was saying, you guys have a lot of really good mushroom emergences when you have rain. If you've ever been to Ireland, it is just one giant rainstorm. And this is another example of a plant pathogen that is around today. Even though you don't hear about it so much in the news, right? That's not causing a mass movement across a continent. It is still something that you can experience in your gardens today, but there's just been a lot of work to prevent it from happening. It's just a cute picture of my cat. But anyway, the potato famine can also infect other members of the Solanaceae family, such as tomatoes. Potatoes, tomatoes, tomatoes, potatoes. But we're still studying this today. And this is just an amazing story of a scientist that is very near and dear to my heart. This is Dr. Mangaligli, who has been working. He still works today. He's 98 years old, and he's still, oh my God, my cat. He still runs to the office. It's insane. He's 98 years old, and he still runs to the lab and does pertinent science. But his whole career has been dedicated to preventing a new variety, a lot like, coronavirus variates, but he works with tomatoes to make new tomatoes that can always keep up with the evolving and adapting plant pathogen. There he is. He's cute. Still working. Awesome guy. And this is probably the piece de la resistance. I don't speak French, but I've heard that before on TV. This is a story of the American chestnut, which there are American chestnuts in California. They've been migrated by humans, but most of the American chestnuts were on the East Coast. And I'm saying they were in past tense because they've been killed by a plant pathogen. And there's a picture here of a little baby cradle and a coffin, because this used to be, here's a California example, the American chestnut was the redwood of the East Coast. And so these don't look like anything exciting to you guys with all your huge trees, but for us on the East Coast, these are huge. And of course we did logging and stuff, but all of these trees mostly died because of a plant pathogen. And you can still find old barns here in the Appalachian Hills that are made out of chestnut because the wood is phenomenal. But like I said, it was all throughout most of Appalachia here. And because of a fungus, so here it is on a Petri plate, this is the Cryphenectria parasitica. This is the ascomycotin fungus that kills trees by causing cankers. So you can see all the orange stroma there that is causing disease on that American chestnut tree. And it totally wiped out. So this red area now is the range of this fungus. It totally wiped out the whole range of American chestnut. And you might find them in the forest today, but they'll look something like this, where they can't even get to an age that they can functionally reproduce, because the fungus will just hit them hard again, hit them hard again, and soon they will probably not be able to even do this. And it's greatly changed the forest composition of the East Coast. So this is my home, a beautiful forest with a lot of shade, a lot of conifers. And it's just an example that if you were to have a pristine forest, here's some, you guys have trilliums over there. You also have some trout lilies. And we have, you know, species that depend on the shade, like these birds and tree fish and stuff. If you get rid of a keystone species like the American chestnut was, you increase the amount of light in your forest, it changes the forest composition, and then it's also a suitable habitat for invasive species to come in and change the whole landscape. And we no longer have our fishies, our birdies, our bears, and all that cute stuff. And there's a lot of science that goes to this. So there's a ton of grant money and all this people working on it, spending their time and trying to fix this problem still today. And these are examples for any kind of plant pathogen that can, like, you know, this can be extended to any kind of species combination, but you can try to fix the problem by breeding the host with a resistant variety. So they'll try to breed an American chestnut with a Chinese chestnut, which is resistant to get certain traits. There's a virus that infects the fungus that they try to control it with. And there's also transgenic chestnuts, GMOs, that are being very successful against this blight. And so my experience in working with chestnut blight a long time ago was that we actually would insert a virus into the fungus. And so I just think it's a really fascinating story, because sometimes we think of these organisms as being so static, right? We identify them in our field guides and like, that's it. Well, there's other things that infect them and interact with them, just like us. So this is Cryphenectria parasitica, the chestnut blight, this beautiful orange fungus. But when you give it a viral infection, it loses its ability to make orange, and it also loses its ability to attack the American chestnut. And the American chestnut then can fight back because the virus is weakened, and it's not as deadly as if it was still orange and fully potent or virulent. And this virus is a lot like viruses with us, like the AIDS virus is transmitted through sexual activity. These fungi will mate and have sex, and they can infect each other with the virus. But fungi don't like everybody. Again, a lot like us. You don't mate with everyone. You find people you like, and then you mate with them. And there's other people where you build a chemical arsenal wall and you say, never, ever come near me. And that's what's going on over here. But this has been a really interesting study where the trees in California look a lot like these trees here. These are trees in Wisconsin. These are American chestnut trees in Wisconsin that are actually full grown and mature and will produce chestnuts. And the infection is really small in these trees because this fungus has a virus. And so instead of this going all around the entire tree, the tree is able to fight back and then only have a scar on its trunk instead of being completely killed. So I told you all of that just because it's the story that I'm familiar with, but really it's also a perfect example of what plant pathology is and what scientists do to combat these diseases. Because a lot of times you'll hear about these diseases and you'll be like, oh no, sudden oak death, that sounds so scary. Like, what am I supposed to do? And you don't ever hear about what you're supposed to do on the news. But there's a lot of research that's going on behind the scenes by a lot of different professionals at different agencies and universities, and there is usually a success story that they're working on. So this is the California example I wanted to focus on because it is of the same caliber, and it's not exactly in the same place as you guys in Southern California, but it is of the same caliber of scary as the American chestnut story. So sudden oak death is caused by a plant pathogen called PHYTOPHTHORA ramorum, and its main host is tan oak, which I was just looking at CalFlora, and tan oak is down in your neck of the woods. So this potentially could be a problem for you guys eventually, or in places where you do have a lot of water, if that happens with irrigation systems, but it's mostly in Northern California and Oregon at this point, but it is spreading. But this is a plant pathogen that causes these cankers here. So these dark oozy sepy points on this tan oak are oozing cankers that are dead spots underneath. So the tree is no longer able to transport water into its canopy, and it will also cause the leaves to die. So this is a tan oak leaf that has an infection and is rotting away. And you can see, this is a photo from the Forest Service out in California. You can easily pick out the trees that had healthy leaves on their canopy, and then all of a sudden they got this infection and they died. So you'll see a dead, crispy canopy in the forest, and it spreads very quickly. But tan oaks aren't the only hosts that suffer from mortality from the sudden oak death causal agent. Sometimes it actually has been found on manzanitas, and I know there's a lot of different species of manzanitas in California, but it's also on your coast live oaks and California black oaks. But all of these will be in different severities. So it's a lot like, if one species is susceptible, actually a sad but good, well understood example is when Europeans came over to the Eastern United States during colonialism, they brought over blankets that had smallpox on them. Well, the Europeans would still get smallpox, but they wouldn't die. But the Native Americans got smallpox and it wiped out their population. So even within similar species or same species, you can have different levels of severity. And that's what's being found on these oaks and also other species like hazelnut, some ornamental maples that are in the cities, and rhododendrons. And this is a really pretty recently discovered disease that's spreading really quickly. You can see 2007, and there's still progress on it being seen today. And there's not as much hardcore science because of the factor of time, like the example of the American chestnut, but there is science and methods that are happening out in California to stop the spread of this plant pathogen. There's no way to cure a tree once it's been infected, and that's usually the same with most plants, but you can prevent this disease from happening. There's also improved early detection methods with genetics. So foresters and people just in general that want to contribute can take samples of the soil and send it into an agency like the Forest Service and look for the DNA of the plant pathogen. And if they find it there, they'll stop all movement of cars, of people there or wood, and then they can slow the spread of the disease. But they'll go in and they'll find a disease and they'll cut out the sick part of the tree and get rid of it. And things that you can do if you ever hear of something like sudden oak death, or really any kind of pathogen or insect that's affecting your forest and causing sicknesses and disease, you can make a difference by not cutting firewood and taking it to a new county. So that is the #1 way that diseases spread, because humans have vehicles that we can drive into the national forest, get covered in mud, and then drive to the next place we're camping next weekend. Even your muddy shoes, if you go in a forest that has a lot of dead trees, you should take them and put them in the tub and clean them with soap, because you could potentially be transporting a plant pathogen or an insect. But the thing about why we're talking about this, not only because it's a plant pathogen, and a lot of them are fungi that cause these diseases, if you lose a host of one of these plant pathogens, there's a possibility that you're going to lose a lot of the things that this club cares about. So in California, you know, Matsutake, which again are more northern, where this disease is being really serious, they're commonly associated with tan oak. So I'm sure all of you guys know what the Matsutake look like, but they are associated with a tree that could be dying, and then you will see less of these beautiful mushrooms that are in a symbiotic relationship with that host plant. And here's a few other fungi in California that are related or like confirmed associated with tan oak. And I'm sure a lot of you know there's so much we don't know about fungi at this time. I bet there are thousands of fungi more that are related to tan oak, and probably some that are incredibly specific. So this image here from CalFlora, which is a great resource if you're not familiar with it, it's for plants, unfortunately not for fungi, but all these blue dots are occurrences of tan oak in California. There are a lot of other important diseases in California. A few that came to my mind right away was this gray mold that will grow on agriculture, and you do have a lot of agriculture down where you are, as well as throughout most of California. White pine blister rust is a fungus that will attack white pines, especially the white bark pine, which is a rare species in California, but is down not too far from you guys in the Southern Sierras down here. And it causes, again, cankers to grow with oozing fungal pustules that will kill entire branches, and it happens in a couple of years. Thousand cankers disease is a fungus that's transmitted by a beetle, which attacks our walnut trees. And you do have a lot of your Juglans hinsii. Here's the map for that tree species that grows around Los Angeles and also San Diego. And of course, our wonderful olive trees and oleanders that fill the streets and neighborhoods of California, they suffer from plant pathogens as well. Bacterial organisms cause leaf scorches, but it can also, is something that we can't see with our naked eye, but we can see the symptoms that this organism causes. And I saved the scariest for last, because, you know, they always say this on like TV and late night shows. Oh God, where would the Californians be without their avocado toast? Everyone eats it, it's fine. But there is a disease of avocados, which has been recently found and is being studied, that is carried by a little tiny beetle. And so this little beetle here carries this fungus in its face. And this is actually a cross section of this beetle's head. So this purpley part here and purple part here, those are the eyes of the beetle. The orange part at the top is the brain. And then this little circular area is the throat, the esophagus. And right here are these little cheeks. They have M's there. These cheeks, a lot like a little chipmunk, are filled with fungal spores of a fungus that this beetle carries around because it eats it. And it eats it after the fungus has eaten a tree. So you see all these separate little circular holes here. Those are all drilled in points from different beetles that took their chubby cheeks, spit out the fungus started eating the tree, and then they eat the tree, or they eat the fungus, because they can't digest the wood, but they can digest the fungus, which can digest the wood. But it's not all doom and gloom. We can use plant pathogens to our advantage, because they can control species we don't like. And this actually happens a lot more common than, you know, what the news tells you, because usually it's scary stuff on the news. But this is a tree called Tree of Heaven, and it's all throughout the US. And you might think it's crazy, but I have actually found it at Red Rock Canyon in Southern California. And it causes the fungus that kills it causes a disease a lot like the tan oaks that we saw earlier, where the leaves will not get water and they'll drop, and the whole entire tree underneath the bark will die. And then the tree dies in a couple of years or even a couple of months. And so if you isolate this fungus, it's just a classic fuzzy white mold that you have to use a microscope to identify it, but it can also live in the soil. So it'll stay in a dormant state in the soil for many, many years. And land managers are actually able to take this fungus when it's living in the soil and distribute it in an area that has a high amount of these invasive species and then the fungus will come out of the soil and infect the tree and control the invasive plant that way. And there's many strains of fungi that have selectivity for their host, because sometimes people get scared when they think about introducing another organism into the environment. But lucky for us, that fungi can be super hyper specific. And an example of a different strain within a species is something like you and I are both Homo sapiens, but you might be lactose intolerant, but I'm not, right? We're still Homo sapiens, but you have genetic framework that makes it that you can't drink milk, but I can. And so fungi will have that ability too with different hosts of plants that they eat. And this is an example of the same fungus, that verticillium that I showed you in the picture before. There was a strain of it, so same species, but different strain. There was a strain from tomato that went on here, and it didn't kill the trees. But there was a strain from Tree of Heaven, and it killed the trees because they were specifically adapted to being able to use their genes to kill the tree. So in conclusion, the field of plant pathology can be super depressing if you only hear about the death and despair of species, but it has a lot of avenues you can follow as a scientist. So maybe you'll get funding in grad school. It's very dynamic and ever-evolving field of study. Something that's really amazing is that some of the things I did during my PhD work were not even possible to anyone 2 years ago, because it was a brand new machine that was able to blow stuff up. The field of plant pathology greatly impacts our lives literally every day. The taxes that come out of your paycheck go to funding plant pathology research and preventative care. And it has the potential to make the world a better place, or sometimes worse. But of course, it's super fun and always interesting. Every time I look in a microscope, my mind is blown. This is all fungi, and these are all their reproductive structures, just being wild. So you guys are in California, and the leading groups that study plant pathogens in your area, it's UC Davis, but it's because UC Davis out of the UC system is the agricultural, mecca of your system. So UC Davis has a Department of Plant Pathology where they study pathogens in your entire state and most of the West Coast. You also have the California Department of Food and Agriculture, who you can send samples to. So if you even have something that looks crazy on your tomatoes, you can call them. They will come out. They will say, oh, this is something new we've never seen before. They'll sample it, and someone will do what I just did and look at things in a microscope and ID it for you. And there's also the UC IPM website, which has a lot of photos and a lot of stuff that you can learn about plant pathogens. But Los Angeles County is really awesome because they even have a county department that handles plant pathogens and has a lab which works closely with the state and closely with the feds. So you get a super expert, really hyper local situation in LA. And that is my talk. Like was said earlier, if you are interested in Instagram or social media, I'm on there with my silly nickname from when I was a kid, K-Dubs, the Hikey Scientist. And if you have any questions via email, that is my email right there, and I would love to hear from you and see some cool stuff. I really appreciate you guys letting me be here. It's always fun to meet new people and see how happy and big a mycology group is. So I'm going to stop sharing, and if you have any questions, go for it, unless there are rules in your club about how that happens.
No, I don't think there is any rules. People can raise their hand.
There's no rules.
Not quite. We had a little Zoom bombing in the past. But I think people can just introduce themselves if they want, or we do use the chat. And if you're having trouble doing the chat, Aaron and/or I will read the chat to you. So I see some thank yous there already. And maybe I'll just start with a question.
Okay.
So for that chestnut fungus and the virus, is the virus spread in the spores?
No, it's mostly spread in the hyphal anastomosis. So hyphal anastomosis is when they merge and mate, and then they swap genetic material. So it's an RNA virus, which is not it's very different and very dependent on the ability of the thing it's infecting.
So how does the virus originally get into the fungus?
So we can inject it, but the easiest way for us to do it is to just have them mate.
And then do you spread the fungus around to try and protect the trees?
Yeah, we call it a slurry. So in scientific papers, it's called a slurry, but it looks just like an orange Julius. And we put it in these big bottles. We use an industrial sized blender, and we just take all of the Petri plates that we grew the fungus on that has a virus, and we dump them all in this big giant blender, blend it up, and then put the slurry. So I'm not sure if everybody's heard about this, but fungi can have, depending on the species, can have many different mating types. So for us, even though, you know, it's not what it all seems in textbooks, right? There's male and female for humans. And so we have technically 2 mating types that pair up and can swap genetic material and create offspring. Well, fungi can have 5 mating types. They can have 10,000 mating types. They can have 20,000 mating types. And so one of the challenges of spreading that virus was figuring out the mating types of Cryphonectria parasitica, the fungus, and who would swap it with who. Because there would be different strains of the chestnut blight fungus that didn't mate with anybody, or what we knew, and so they would still be really aggressive in the forest. And we were trying to figure out who they wanted to mate with, so that way we could give them the virus.
I see. Thank you.
Yeah, it's wild. Thank you for a good question.
Someone else? I see Andre has his hand raised. Go ahead, Andre, you have to unmute.
Yeah, it sounds like it's a very recent doom and gloom. So is there historical evidence of these pathogens, or is it really something very recent? So could we attribute it to like human pollution or something, that these pathogens have really, you know, made such great headway, I guess to say?
It's more so, I mean, it's a lot of different factors. It's the spreading of, the biggest thing in my personal opinion, based off of my professional experience, is the swapping of material all over the world, right? So the chestnut blight came from a Chinese chestnut that was planted in New York City. So we didn't have that hundreds and hundreds of years ago when we weren't taking boats full of plants and their diseases to new locations, because the American chestnut evolved to be resistant to the things in its own American ecosystem. And when you introduce a new, you know, species that's similar enough that it's co-evolved pathogens that it doesn't care about, that it just goes onto the free buffet of the American species. And that's what's happening a lot. A lot of these pathogens come from other countries where they are co-evolved and not as serious on other organisms in their native range. And then they come over here and there's no genetic resistance or even recognition, and it's just wild. Another thing that I can contribute it to is climate change and the warming of just the environment and just the changes in the weather pattern. That's one, stressing out the plant that's its host. And then the other scenario within the climate change aspect is that we're creating a more suitable environment for microorganisms, because they like it when it is wet, and they like it when it's random, and they like it when it's, you know, more humid. So it's we're creating a better environment for pathogens to infect hosts that are getting stressed out.
All right. Well, thank you so much.
Yes, thank you for a very good question.
I see Ben Snyder raised his hand, and then Kate after that.
Yeah, thanks. I actually have a question about one of the first things you talked about. Do you think there's any particular reason why most of the fungi with parasitic habit in like a pathogenic way are ascomycetes? Is there some sort of like causation there, or is that just an interesting fact?
I think it was a diversion evolution of function. So, I mean, they're both ascomycotin and Basidium mycotin are phylum within kingdom fungi, and that a main feature of separating them is their ability to make associations with their host or their plant associates.
But in a parasitic, like a pathogenic way, you mean?
Yeah, but I mean, I think that function or form of the mushrooms that we see is kind of a result of their mostly under the radar relationship with plants. So they'll be intertwined the whole time and they won't be out on the big scene, but then because of their evolution, which is different than constantly trying to eat, kill, they make a mushroom once and then they're done. Whereas the ascomycotins, it's just a product of their evolution where they've continuously evolved to not have to rely on that. And so their relationship is just, we're just going to make really small spore structures that will infect and kill. And then they make their own genes, like, you know, continuously through time, they continuously diverge. So that way the basidiomycetes are like, oh, I'm going to make genes or genetic material code that allows me to associate with this tree species versus this one, whereas this one's like, I'm just going to eat everything. And then they just continuously get more and more different.
Yeah. I mean, because there are parasitic basidiomycetes as well. We have someone in the chat here talking about some Ganodermas taking down their ash trees. So that's what that was interesting. I guess.
Yes. Yep. And I mean, there are probably some guys that are just right in the middle of that main delineating feature. So for the most part, I mean, the Ganodermas and the Felinus, the polypores, yeah, they act as these like secondary pathogens that usually they're in their own bracket. You know, they're kind of like I don't even know sports. They would be like your local baseball team versus the Mets, you know? So I don't know sports.
Kate, you had a question?
Yeah. Hi.
It's really nice
So
to see
happy
you.
to have you.
Yes, thank you.
Yeah, a question about pigment, because I'm always going to go to aesthetics first. Do we know why the virus that you mentioned that affects the orange color, do we know why it loses its pigment during a parasitic cycle?
So that's not always the case, but in that certain environment, I mean, I hate to think of it like AIDS, just because AIDS is such a sad disease to think about, but it's a lot like AIDS. And when you're immunocompromised, your body's just trying to live. And so if you are making pigments, usually pigments are a result of they're a secondary metabolite. And so it's usually something that's not necessary. So the fungus is no longer doing all the functions of all of its normal things to just be happy and healthy. It's now to a point where I just need to live. And so it no longer makes the metabolite, which would make the pigment.
That makes absolute sense. Thank you. Yeah, I was thinking it could be related to function. So
yeah, but it's not always the case though. Mm-hmm. So it's different for every single severity of a disease or, you know, whatever's going on.
Yeah.
We're just super lucky that there was a way. And actually, to get into the weeds of that whole study, there was a fungus that, not my lab, but a lab that we worked really closely with, and we like utilized the results of their study. They made a fungus, it's genetically altered. So like we actually went in there and figured it out, and we called it the super donor. And so the super donor was able to mate with almost everybody, and its pigment, when we found it out in the field, we knew what it was because it was brown. So it was just at the point where it was healthy enough to make a slight coloration and not be white, but it was brown, which made it still stick out from being orange.
Very interesting. Thank you.
Yeah, thanks.
Victoria? You might have to unmute. Still not hearing you.
Maybe it's connected to a speaker or something, or headphones.
Is that any better?
Yes.
Yeah.
Okay, all right, cool. So I had a question about the beetles that were affecting avocado trees. At one point I was reading something about another parasitic this was specifically about nematodes, but it only was able to infect the host tree if it was present at a certain stage of its life cycle, of the tree's life cycle. Is that, do the beetles affect the avocado trees at a similar, like, is it only if it's within a certain time frame of the avocado tree's life?
They might go for a feature of the beetle and their reproductive strategy is that they burrow inside of the tree, right? So if we take the tree and we go like this and we look at a cookie, like the horizontal crosscut of it, the beetle, as an ambrosia beetle, comes in and drills holes in this space and fills that with fungus so it can eat it. And so if there isn't a wide enough tree, which is a function of its age, right? Because they grow like this, if it's not old enough to have enough real estate for them to drill into and bore the fungal holes, then they won't infect it. So that's why you'll see things like really young orchards don't have a problem, but then when they get older, they've reached this function of being adequate for an insect vector to infect them. And also trees, they have lifespans just like us. Sometimes it can be 3,000 years old, like the sequoias, or sometimes it can really only be 30 years old, like a lot of the ornamental trees or orchard trees, like apples, that, you know, the quality just goes really far down because of the lifespan of the tree. So the trees do not have immune systems, but they do have response rates to infection. And as they get older, they can't signify all the functions to kick beetles or fungi out. So that goes back to the whole thing of when I said it with sudden oak death. If a plant gets a disease, it's like, it's done. Like you can like cut off a limb, but this limb will never get better because it doesn't have an immune system. The way that plants protect themselves from disease is sacrificing. So they'll like drop a branch because it had a pathogen in it. They'll like block off all this and they'll drop the branch. Thank you.
Aaron?
Trees can be smart. Thank you for your talk. And I'd love to hear you talk about entomopathic fungi at some point. But my question is, what are some of your favorite ways of doing science communication and outreach regarding these difficult topics of plant pathogenic education and to the public and whatnot?
I really like the live feature, not the live feature. I like the story feature of Instagram because, you know, it's more than a post and you can watch it all in like one go and like see with real life examples. And honestly, I've been thinking a lot lately, like with my new job, I'm doing something that's so different than what I used to do. And I don't have a lab. I'm building a lab. So I can't even like show how to do a lot of the stuff I used to do and I miss it. But eventually I will have a lab or I'll have a new job. So who knows what'll happen, but I really enjoy showing in step-by-step what it actually looks like because you can only get so much from a photo, right? And I do like making the photo posts because I like it. And that's most of my experience with Instagram. I only do things if I like it. I'm not going to become a TikTok influencer. It's too much. But, you know, I just feel like showing things you're genuinely interested in and you want other people to know about and not treating it like it's a formula. I mean, I think genuineness and then also taking things down and showing them step by step is really what has worked for me.
Very cool. That's really helpful, actually. Thank you.
Yeah, don't do anything you don't want to. Like, don't do these weird like emails to sell, I don't know, an ornament that looks like a mushroom that you don't care about, but you're like going to get a shout out. Don't do that because it's not real, right? You don't care about it. Just show people genuinely what you're interested in, and then someone will be like, whoa, that's cool.
Totally, choose.
So I see a question in the chat from Lynn Lineman. She says that Gandodermas are taking down one ash tree after another in my neighborhood. What can be done?
So there's a couple of things. I'm gonna suggest that you contact your county extension agency so that Los Angeles County Plant Pathology Lab, they can probably help you. I would think, even though I'm not an expert on the West Coast, Ganodermas usually show up when there's something else that's already been wrong. The ash trees, I don't know about you guys. I don't think you have emerald ash borer yet, which is an insect that kills them. But there could be something like that's really stressing them out as being street trees in your neighborhood. And then they got these infections of the Ganoderma. So there's usually nothing you can do because, again, trees don't have immune systems. So once they're showing the fruiting body, which is the big Ganoderma conch, if your tree has the big conchs on it, it means that it's, you know, infiltrated enough on the inside with fungal mycelium that they've eaten enough of the tree to make up reproductive structure and reinfect other trees. It might be worthwhile to remove them and then plant something else that isn't going to be susceptible to the Ganoderma. And the county extension officer will definitely help you. I mean, that's their job. You're paying them to do that. And you might even make a friend. They'd probably be excited that you knew it was a Ganoderma.
It's true. Do we have any other questions?
Oh, thank you for doing that shout out, Kate. That's very nice of you.
Excellent.
It's a fantastic episode. If nobody's listened to this specific one, it's so interesting. And I've listened to a lot of the episodes, and the way that like Allie gets excited about this information that, you know, is something that we don't hear a lot about is just, it's pretty rad. And it makes you, it just turns, like, shines a light onto this thing that, yeah, we normally feels depressing when we're walking into the forest and we're seeing, you know, scars, and it kind of like turned my perspective around a little bit, and I feel a lot more hopeful now after seeing a lot of Kristen's posts as well. So
thank you.
You're so welcome.
Also, there are a lot of native pathogens too, right? And it's not always like a scary thing if you see like a weird mark on a tree. It might be something, you know, just like we get athlete's foot, right? I'm not dying because I had athlete's foot 5 years ago. Don't have it anymore. No, I'm kidding. I don't know. But yeah. So there are native pathogens that can be really interesting and beautiful in their own way, because they are also organisms that are just trying to reproduce and live and thrive. But unfortunately, there are the scary ones that get our attention because of they're invasive and they are not here. So our plants are not co-adapted to survive them. I'm not being rude. I'm answering a question. I'm turning around to get a book to answer one of the questions. So the question was, are there any really good plant pathology books? And there are. Here's a good one that is across the whole country. It might look dated because it is, but it's one of the best. It's what I used to study every page for my PhD. It is Diseases of Trees and Shrubs, and it's by Sinclair. And it has, oh look, there's a secret book on the inside. And it has awesome pictures of a lot what we talked about. So there are some pictures of the symptoms, and then it usually includes pictures of the actual organism too, especially with species that you have to look at the reproductive structures to identify. And this is across the whole country. So it'll apply to you over there, and it'll apply to me over here. Another book is by Agaros, and there it is. So this is a really good book for studying plant pathology. Again, I read this from both my master's and my PhD. This is Plant Pathology by George Agrios. And it's awesome. It is pretty current. I mean, it's a textbook. So if you like reading that, it's got life cycles and many different pages of different stuff. But it's got some really good diagrams, and it can be fun. It helped me get my degree. And here I am now. Have you ever used a companion infected plant to help control an invasive species, like how bayberry spreads a killer fungus to wheat? No, I have not directly done that, but I did do a side project because I love that component of plant pathogens. So I was distributing a native plant pathogen that was killing an exotic species, and nobody knew what it was on beforehand. And we honestly still don't. And I kept wanting to be like, I'm going to find it in the woods. I'm going to find some weird little horse nettle herbaceous plant that's slightly related to the exotic species. And then it will be where I find the fungus originated from, acting as a nothing like pathogen, right? The athlete's foot scenario. And I never found it. I still always look, but maybe someday. But that is a really cool component, and I'm glad that you asked about it. DRD.
I think that's David Roman Daniels.
How did I know it was native? Because of genetic studies. So we described the genome and compared it to other genomes that were known within the same genus and in host relations, and looked at phylogenetic trees that we originated, and also did host severity or pathogenicity tests. So we tested over 300 species of plants to see what it would infect. And a lot of concepts led us to believe that it was native, especially the factor of looking at the genome. There you go, nice, someone shared it. Thank you, Dan Lopez. Yes, bay laurel is thought to be a host for the sudden oak death. And I'm sure different species or different varieties even of that will be affected differently. Nice cat butt over there, Vanessa. They gotta know. My favorite mushroom, man, it really changes all the time, especially with the seasons. And also, you know, I'm sure you know categories, right? Favorite edible or favorite, oh God, only one. I know, I like Heriachromipes, if that counts. That's just, I like it a lot. I don't know if I'll be able to find it, but you were talking about boletes in your identification. It's a beautiful little pink bolete that has a chrome yellow base. So this is a fun book on the East Coast. You're talking about your bolete rules. On the East Coast, there are hundreds of boletes, and you know they will have features that are like, does it stain blue in 1 second or does it stain blue in 30 seconds? And then it's a whole, that's the only thing that's different. So I really am enjoying the boletes. I like Heriachromipes. It's a really cute little pink thing. It makes me think of my niece because she would always ask me if I found any pink mushrooms, and she wouldn't care if it wasn't pink. Mm-hmm. So that makes me think of her. But that was a good question. Thank you. And thank you for finding the books. That was really nice. It makes everything a lot easier.
Do we have some additional questions? Well, I think we have your contact information. And anyway, I'd like to thank you again for a really interesting and stimulating talk. See one more. No,
that was me. I was just sharing a Wikipedia page for the pink bolete. Okay. Thank you very much. Yeah, I had
well, thanks again. Really appreciate it. Thank you for coming.
Of course.
Zooming.
Yeah, I hope to, you know, when the world goes back to normal, I hope to return to traveling, and I would love to come to a real life meeting and see your guys' mushrooms and stuff. That'd be awesome. So thank you so much for thinking so highly of me. That means a lot. And I'll see you guys hopefully later.
Thank you.
Thanks again.
Bye.
Good night, everyone.
Bye.
Bye now.